Hello, I'm Dr. Joe Keating. This is the third and final lecture of How do you solve a problem like morphology? Last time, we talked about empirical and simulation approaches for assessing the performance of different morphological phylogenetic methods. In this lecture, we will consider more broadly what we know about morphological characters, how they evolve, and how we can model their evolution more realistically. Unless you have accidentally signed up to the wrong course, you probably recognise these two gentlemen. Charles Darwin and Alfred Russel Wallace were both talented comparative anatomists, and through studying the morphology of animals and plants, both independently came to the same startling conclusion: that life on Earth had evolved. Darwin and Wallace provided a compelling mechanism to explain evolution: natural selection. Their insight was that populations exhibit variable phenotypes. Because of this, some individuals within those populations have better chances of surviving, reproducing, and passing their advantageous traits on to the next generation. The idea is summarized by the phrase, survival of the fittest. Unfortunately, morphology evolves over such long timescales that we cannot conduct empirical experiments to observe it happening. However, there is abundant evidence that corroborate Darwin and Wallace's hypothesis. Over short timescales, we can observe how natural selection affects population structure when environments change. The fossil record provides a long-term perspective and reveals how morphology is transformed as organisms transition between ecological niches. It is evident that over macroevolutionary timescales, selection plays an important role in driving morphological change. Natural selection remained the predominant explanation of evolutionary change until the 1960s, when molecular data became increasingly available. Independent experiments conducted by Kimura, and also King and Jukes, led to the surprising conclusion that molecular evolution is a stochastic or random process. Their results suggested that most molecular sequences evolve through long-term genetic drift, and that substitutions are selectively neutral. This became known as the neutral theory of molecular evolution. Our best models of molecular evolution are informed by the neutral theory and make some important assumptions about character state changes based on the expectation that such changes are selectively neutral. The MK model of morphological evolution is derived from the Jukes-Cantor molecular substitution model. Consequently, it also makes assumptions about the neutrality of character state changes. This raises some interesting questions. Is the MK model appropriate for morphological data? And, if not, does this matter? Myself, with colleagues from the University of Manchester, designed a simulation experiment to address these questions. We generated simulated datasets using two evolutionary models. Unlike previous studies, these models do not require a fixed tree. Instead, the tree is an emergent property of the model. Let's first look at our stochastic model. MBL 2017 is a lineage model. It starts with a single lineage with a random binary morphology. At each iteration, there is a random chance that a character state change will occur. There is also a random chance that a lineage will speciate, i.e. be replaced by two daughter lineages. And finally, there is a separate random chance that a lineage will become extinct. In this way, the model can simulate the stochastic evolution of organisms through time. When the simulation ends, it outputs the simulated tree and the simulated morphology of the taxa at the end of each branch. Now let's look at the selection model. Trivosim operates at the level of individuals. It starts with a population of individuals that share a random binary morphology. There is also a simulated environment composed of random binary strings. We can measure the fitness of an organism by comparing its simulated morphology with the simulated environment. The chance that an organism will reproduce is dictated by its fitness. In this way, Trivosim is analogous to natural selection. At each iteration, each individual has a chance of incurring a random character change. New species are defined by character differences. Similarly, at each iteration, the environment has the chance of incurring a random change. As the environment changes, species that fit the new environment reproduce preferentially, while those that do not closely fit the environment may become extinct. Again, when the simulation ends, we output the simulated tree and the simulated morphology of the terminal taxa. Under a stochastic model, we should expect evolution to be predominantly divergent. Lineages will accumulate independent character changes as time passes. Consequently, we should expect to see strong morphological coherence. That is, a strong correlation between the phylogenetic distance of any two taxa and their morphological difference. For taxa that are closely related, we expect to observe few morphological differences. However, as the phylogenetic distance between taxa increases, we expect the number of morphological differences should also increase. If the rate of evolution is low, the correlation between phylogenetic distance and morphological difference should be close to linear. However, if evolutionary rate is increased, the correlation becomes nonlinear. Sure enough, data simulated under the stochastic model shows an expected pattern of morphological coherence, suggesting that evolution is divergent. However, data simulated under the selection model shows a very different pattern. Importantly, there are taxon pairs that occupy the top left of these plots, indicating these taxa are very distantly related, yet have very few morphological differences. We would be very unlikely to see this pattern if evolution were purely divergent. However, this pattern is easily explained by convergent or parallel evolution. Under these circumstances, the same characters evolve independently on separate branches. Parallel evolution occurs when branches have similar ancestral conditions, while convergent evolution occurs when branches have different ancestral conditions. Parallel or convergent evolution can occur as lineages independently evolve adaptations to a similar ecological niche. Insectivorous mammals seem to be particularly prone to convergent evolution. However, examples of convergence are ubiquitous across the tree of life, and it is clearly an important mode of morphological evolution. As we have seen, stochastic and selective evolutionary models produce data with different properties. How does this affect the performance of different methods of morphological phylogenetic estimation? In order to find out, we simulated morphological datasets using the stochastic and selection models and estimated phylogeny using Bayesian inference, equal weighting parsimony, and implied weighting parsimony. We found that Bayesian inference was more accurate but less precise than standard parsimony trees, regardless of how the data were simulated. This is similar to the results of previous studies. These results suggest that, even though the MK model is not really appropriate for analysing data that has evolved under selection, it can still be used to estimate trees with reasonable accuracy, which is reassuring. However, the most interesting result was comparing the accuracy measures obtained using bipartitions to those obtained using quartets. For the stochastic model, there was very little difference. However, for the selection model, there was a large difference. Accuracy was much lower when measured using bipartitions. As we saw in the last lecture, bipartition metrics, for example, Robinson-Folds distance, are sensitive to rogue taxa. A single branch that is resolved differently in two trees can significantly lower the number of bipartitions they share. In contrast, the quartet metric is less biased by rogue taxa. The difference we observe between bipartition and quartet metrics suggests that rogue taxa are prevalent in datasets simulated under selection. Furthermore, our results suggest all phylogenetic methods struggle to place these taxa accurately in the tree. Another difficulty in modelling morphological evolution is the nature of the characters themselves. DNA bases, or amino acids, evolve largely independently. However, morphological characters are frequently linked. Take, for example, these stem tetrapod skulls. We could code the variation we observe in a series of discrete characters, each describing the shape of individual skull bones. However, due to the functional constraints acting on the skull, changing the shape of any of the skull bones will require simultaneous changes to adjacent skull bones. The links between morphological characters can be subtle. A good example of this is serially homologous structures, such as teeth, vertebrae, or arthropod segments. In all these cases, the morphology of the individual units are controlled by the differential expression of just a few genes along body axes. While we could code the morphology of individual elements as discrete characters, the individual elements do not evolve in isolation. Perhaps the most frequently encountered type of linked characters are those that are hierarchically nested. Take these lizards, for example. We can capture the variation they exhibit in two discrete characters, the absence or presence of a tail and the colour of that tail. However, we can immediately see a problem with this solution. Observing tail colour is contingent upon the taxon possessing a tail. What colour do we code for a tailless lizard? We can represent contingency diagrammatically. Tail colour, character 2, is contingent upon tail presence or absence, character 1. This is a fairly simple contingency, but they can be more complicated. Character contingencies can be stepwise. In this example, character 3 is contingent on character 2, which, in turn, is contingent on character 1. Two or more characters may be contingent on a single important character. In this example, character 2 and 3 are independent of one another, but both are contingent on character 1. This kind of contingency can occur after the evolution of a key innovation, for example, the evolution of the neural crest in vertebrates. Alternatively, a single character may be contingent on two or more independent characters. In this example, character 3 is contingent on a particular state of character 1 and a particular state of character 2. These character hierarchies reflect something deeper. The relationship between discrete characters reflects the gene regulatory networks that control the development of traits. The evolution of gene regulatory networks is a hidden process, in the sense that we do not observe it when studying morphological traits and constructing discrete characters. However, failure to account for this hidden process can result in undesirable behavior when trying to reconstruct character evolution. In a 2019 paper, Sergei Tarasov presented an innovative solution to this problem using structured Markov models. These models allow us to consider both observable morphological states as well as the hidden gene regulatory states that underlay them. For the tail problem, we can amalgamate the two discrete characters into a single structured Markov model with two possible solutions. The first allows tail colour to evolve independently of tail presence, while the second requires that tail colour evolves via a switch-on dependency. Put simply, tail colour cannot evolve if a tail is absent. Using a simulation approach, Tarasov showed that these structured Markov models are less biased by hierarchical characters than conventional methods such as parsimony or probabilistic estimation using the MK model. Structured Markov models offer an intuitive way of modelling hierarchical morphological characters and allow us to incorporate knowledge of gene regulation. These models break down the distinction between character and state and suggest that, in the future, complex morphological structures, or perhaps even entire phenotypes, could be encoded in a single structured Markov model. So, to summarise, in this lecture we talked about some of the difficulties in modelling realistic morphological evolution. Unlike molecular evolution, morphology probably evolves via selection, at least on a macroevolutionary scale. Selection can lead to parallel or convergent evolution. These processes lower the coherence of morphological data and can lead to rogue taxa that are difficult to place regardless of the method we use to estimate phylogeny. Another difficulty is that morphological characters are frequently non-independent. Characters may be hierarchically linked due to underlying gene regulatory networks. Conventional methods can provide undesirable solutions when characters are hierarchically related. However, structured Markov models allow us to model the evolution of both hierarchical characters and the hidden gene regulatory networks underlying them. You can find further information about modelling morphological evolution in these references. Hopefully, these 3 lectures have provided a good introduction of how we estimate phylogeny using morphological data, how we can measure the performance of different methods using empirical and simulation studies, and some of the difficulties in modelling morphological evolution more generally. If you have any questions about any of the lectures on morphology, drop me an email at the address below.